Hey everyone, welcome to the Timposium. So I'm Tom Shenk, a friend of today's honoree and a member of the Hepatitis B Foundation's Board of Directors. I'll start by saying something that we all know. We all know it really well. Tim Block and Joan Block are one ultimate power couple. We're all fortunate to know them. And today, we celebrate Tim Block. As we all know, Tim is a co-founder and president of the Hepatitis B Foundation, which is the world's preeminent authority on hepatitis B education, advocacy, and public health research. He's a co-founder and president of the Baruch S. Blumberg Institute, whose discovery science has helped to build the foundation for therapies currently on the market, as well as multiple therapies in the pipeline for hepatitis B and liver cancer. And Tim's a founder, president, and CEO of the Pennsylvania Biotech Center. The center got its start in an empty warehouse on this site, and it is now home to 49 companies that contribute importantly to the development of new diagnostics, therapeutics, and the local economy. Today, we're gathered in the center's newest state-of-the-art building. The Hepatitis B Foundation, the Blumberg Institute, the PA Biotech Center. How could one person create and build all of that excellence? I first met Tim in the mid-1980s when he was a freshly minted assistant professor at Thomas Jefferson University School of Medicine. I actually remember sitting in your office, Tim, and talking about your work. And that's really saying something, because these days I don't remember half the meetings I had just last week. But I can tell you that I remember talking about your work and really being impressed by how you thought about things. And so I thought that this guy is really thoughtful and is creative. So we kept in touch through the years, occasionally meeting for dinner halfway between Princeton and Doylestown. I joined the scientific advisory board of the, and I can never remember the name of this institute. It's the Institute for Viral Hepatitis Research, the IVHR, which is now the Blumberg Institute. And finally, Tim invited me to join the foundation's board, an invitation that I was very pleased to accept. And I've grown ever more impressed as I've watched this campus flourish through the years under your leadership. So now let's watch a video message from the honorary chair of the Timposium, Raymond Dweck. Raymond is a professor at the University of Oxford, a fellow of the Royal Society, and a friend of Tim Block.
Good afternoon, everyone. Welcome to this Timposium, in which we celebrate Tim's 30 years of contributions in advancing research and drug discovery for hepatitis B and liver cancer. The Hepatitis B Foundation, which he co-founded, has had an amazing impact through its advocacy, education, outreach programs, and research on so many people around the world. The Blumberg Institute, which Tim founded in 2003, has become the center of research in hepatitis B and liver diseases. The property on which the Hepatitis B Foundation, the Blumberg Institute, and Pennsylvania Biotechnology Center reside together in Doylestown, Pennsylvania, will from today be officially named as the Timothy M. Block Research Campus. There will be established the Timothy M. Block Emerging Scientist Fellowships. These honors are so well merited, they bring joy to all of us here, and indeed all of Tim's friends throughout the world. Tim is a remarkable person. As a scientist, he is first class. As a leader, he is scholarly. And as a friend, he radiates warmth and affection. His kindness and sensitivity, together with his humor, makes him an exceptional man who is universally liked. He combines intellectual and personal honesty, professional integrity, with administrative skills and genuine altruism towards others. Of course, Joan, his wife, has played an important part in supporting Tim and helping with his mission, which has helped so many people from around the world find and seek support from the community embodied in the brilliant website of the Hepatitis B Foundation. All your friends in Oxford wish you well in your retirement from the presidency of the Hepatitis B Foundation and also the Blumberg Institute. Tim, you leave a remarkable legacy. May the sun always shine on you. And may everybody enjoy the symposium and know that we are in the presence of a truly remarkable person. Thank you.
Tim, you leave a remarkable legacy, and may the sun always shine on you. Wow, that's heartfelt praise from Raymond Burke. So I was trying to think of how you sum up Tim in just one sentence. And I think that Gene Holmes, the Foundation's VP, VIP as well. For institutional advancement, captured the essence of Tim when she wrote, and I quote, "Timothy Block has a scientist's acumen, an entrepreneur's spirit, and a philanthropist's heart." A perfect description of the mentor, colleague, and friend we honor today. Tim, please stand up and be recognized. Okay, now we're going to enjoy presentations from Tim's former and current colleagues. And Ellen Egan from Worcester State University and Tim's first graduate student will introduce the first speaker. Ellen, the podium's yours.
Okay, thank you. I'm just so delighted to be here today to see Tim and Joan and so many that went through his lab. Thank you for the introduction, Dr. Shenk. It's just a pleasure to be here to share this event to honor Tim today and have the opportunity to see some of the members of his original herpes virus lab at Thomas Jefferson. Let me just say that as a student, Tim was truly a wonderful, supportive mentor. He recognized the potential of those in his lab, and he really encouraged their career goals. And some are here today, Mike and Rob and others, and some that were in his lab that he also recognized as being having a spirit to go into science and medicine and are now physicians in their own clinical practice. Tim, I just want to say thank you so much for giving me the opportunity to be part of your lab and to learn from you. Again, an amazing, amazing mentor. I do want to introduce my friend, colleague, who was in the lab at the same time I was a graduate student, Dr. Mike McIntosh. He began his career in Tim's lab as a technician, as did some others. And within a short time, he became an integral part of the herpes virus group, working primarily on herpes virus latency. Mike then went on to earn his PhD from the now Drexel University College of Medicine, where he studied malarial parasites and provided the first sequence of the mitochondrial genome of Plasmodium vivax. His postdoctoral training took him back to Jefferson to study polyoma viruses, and then he returned to Drexel Medical College to continue work on malaria, where he eventually joined the faculty. In 2001, he moved his faculty to Yale University School of Medicine, where he made discoveries in the field of malaria, which included mechanisms of endocytosis, a new protein trafficking pathway, and a new peroxisome-like organelle. He then joined the USDA's Foreign Animal Disease Diagnostic Lab on Plum Island, where he served in the diagnosis of foreign and emerging diseases in animals. His section deployed new tests for foreign animal diseases to veterinary diagnostic labs throughout the US and abroad, and developed non-biased microarray and next-generation sequence-based diagnostics that led to the discovery that domestic pigs serve as a reservoir for Ebola virus. In 2017, he joined the University of Florida, where his lab now conducts research on emerging viruses and viral causes of cancer. I am just so happy to introduce Dr. Michael McIntosh, who will speak on the block recipe for discovery. So,
well, and thank you everybody for inviting me to speak here today. I would not have missed it. I'm absolutely so proud to be able to talk about how wonderful it is. So, without further ado, the block recipe for discovery. So, and these are lessons that I've learned at the very start of my career. So I was hired by Tim as a technician, pretty much right out of my undergraduate at Penn State. I had one job before that, which was like a 6-month job at a chemical company, and Tim hired me, no research experience, nothing. And he literally taught me how to pipette. Well, and actually, this is how I pipetted in the job before I joined Tim's lab. Really. So Tim taught me from the beginning, although I did learn how to mouth pipette by somebody else, but I learned how to use my thumb in your lab. So that was the first ingredient. So
tell me next slide.
can we go to the previous slide? All right, work systematically. So that was the first ingredient. Next slide. So when in doubt, mutate it. So I can remember Tim telling me every mutant virus you'll learn something the same as the old one. Anyway, and he was right. So you mutate. So I did manage to publish a paper with Tim, and we mutated this latency associated transcript region, and we learned that it really had no function. So which actually became of quite a lot of significance, because it was actually the microRNAs encoded by the intron that actually have the function in latency, in reactivation and latency. So it actually, we did learn something. When in doubt, mutate. So next ingredient, think creatively about impactful questions. And this one is a really important ingredient. When I was in Tim's lab, he had many projects going on. A few that I can remember were, of course, reactivation of alpha herpes viruses, HSV1 Evrys, and DPhil was working on Evrys, and exploiting HSV1 as a gene therapy vector. And this was before gene therapy was even a field. And developing a drug screening platform for protease inhibitors. And this was at the height of the HIV pandemic, we had AZT, there was a drug, but and it had just been approved, but there was already resistance. So new drugs were needed. And as it turned out, protease inhibitors were what were needed to get us out of that pandemic. So this actually got me through graduate school at Anand University, postdoctoral fellowship at Drexel, well, now Drexel University, and junior faculty position at Yale. So this was about a decade of work, and then I found myself literally on an island diagnosing organ and emerging animal diseases. So this was with the U.S. Department of Agriculture and Department of Homeland Security. And so I spent 13 years on this island. But I thought back to what one of the main ingredients, right? Think creatively about impactful questions. And so there's an estimated 75% of new or emerging diseases are actually, they emerge from animals. And so, and our mission was to identify these things and prevent them. But what we ended up doing was we would rule out known viruses, but our freezers were full of unsolved cases, because once we ruled something out, we said, okay, animal trade is open again, everything is great, because it's not any of these known terrible viruses. So my postdoctoral fellow, Rodson Barrett, and I decided we would make a pan-viral microarray, such that we target the most conserved region of all known viruses, and we can identify any virus, whether known or unknown, as long as it's related to something. And this turned out to be an incredibly powerful tool. And as an example, in 2008, Bureau of Animal Industries in the Philippines contacted the USDA to help with an emerging disease in pigs. It was killing thousands of pigs from a respiratory disease and hemorrhagic disease, which they couldn't identify. Their concern was a porcine reproductive respiratory syndrome virus. They had multiple farms with simultaneous outbreaks throughout the main island of Luzon, which is shown there. And when we put it on the microarray, out of 15,000 features to detect different viruses, all of the features for Ebola arrested lit up, and those are shown in green, as well as a few of the features for Ebola virus. So you can imagine we were pretty worried, shocked and dismayed. And after sequence characterization, this really ended up confirming that domestic pigs are actually a host for Ebola virus as well, not just Ebola arrested, but the other Ebola viruses as well, because it was followed up by the animal industries in Canada, their BSL-3 and BSL-4 animals. So another ingredient, add levity. Tim has always mixed humor and science, and I can remember you inviting a speaker to talk about telomere shortening. You remember this one? And you asked, is this why we all get shorter as we get older?
That's funny.
And so it wasn't until later in my career that I asked about apoptosis, which is programmed cell death, is why we have so many great scientists that lose their hair. Always have something else to think about during a conversation. So, and everybody who knows Tim knows he's always thinking about more than one thing at a time. And it's a very important ingredient in this stuff. And you never know when you're going to think something that's important or when something critical that you forgot. So can't leave that one out. And finally, touch as many lives as possible. And Tim definitely touched my life. I wouldn't be the scientist that I am without you. And this institute and the foundation are such a testament, and everyone here today is a testament to how many lives they touch. So before I end, I want to give a quick short story in discovery using the ingredients that Tim has taught. I'm now at the University of Florida. My lab studies viruses in the context of immunocompromised cancer. And I'm actually working on a cancer-causing herpes virus, Epstein-Barr virus. So I've gone full circle from starting my career with herpes viruses to now working on them. So but this story actually starts out with SARS-CoV-2, because my wife, who's also here today, Mary McIntosh, was actually looking at SARS-CoV-2 and mechanisms by which it activates the inflammatory pathway. So, and when we were looking at that, we got interested in ion channels. And so a little bit about ion homeostasis. The sodium-potassium gradient at the plasma membrane of cells, with this high sodium outside and high potassium inside a cell, is one of the most profound biophysical properties of living cells. It drives all sorts of reactions, including action potentials in neurons. And viruses have evolved mechanisms to exploit this and to facilitate their own replication and pathogenesis. In fact, many viruses will encode their own ion channels, and these are known as viroformers. And as it turns out, we actually found that SARS-CoV-2 and actually beta coronaviruses in general encode a potassium channel that causes potassium efflux. This facilitates virus egress, and it also activates the inflammasome. So I asked the question, might Epstein-Barr virus, which is another envelope virus, even though it's a DNA virus, quite different from coronaviruses, might it also use or manipulate potassium membrane potential in the way it undergoes its lytic virus replication? So to do this, and we have to recall that Epstein-Barr virus, like other herpes viruses, has a biphasic life cycle. It has latency as well as a lytic phase. And in Burkitt lymphoma cells that are infected with Epstein-Barr virus, we can induce them into the lytic phase with a histone deacetylase inhibitor, in this case sodium butyrate. And so we use these EBV-infected BL cells, and we developed a flow cytometer assay to measure potassium transport. And that shows, yeah. And so using valinomycin, we can see there's a shift in fluorescence to the right that indicates potassium transport. And so looking at EBV-infected Burkitt lymphoma cells induced into the lytic phase, throughout the course of the lytic phase, where we have virus release at 72 hours, we start to see an increase in potassium transport at the plasma membrane. So we asked if this actually has any consequence on virus replication. So doing the same experiment where you induce EBV infected Burkitt lymphoma cells into the lytic phase, we see an increase in lytic genome copies, and these are inside the cell. And we can actually block potassium efflux by raising potassium, the extracellular concentration of potassium, from 5 to 50 millimolar. And that has absolutely no effect on EBV's genome replication at all. However, when we look in the medium for released virus, we see a dramatic effect. So blocking potassium efflux blocks virus release. So this guy is really intrigued. So we did an informatics search to see if perhaps Epstein-Barr virus might encode its own ion channel. So we looked at EBV membrane proteins, and we actually found some similarity between the HIV's viral protein U, which is the cationic channel, and Epstein-Barr virus's membrane protein 1. So this is an extremely well-studied protein. It's been studied for over 35 years. It's the most potent oncoprotein encoded by EBV. In fact, it can cause tumors in mice just by introducing this protein by itself. It's a 6-transmembrane standard protein, and all of the functions, however, all of the functions, however, have been ascribed to its cytoplasmic tail. And that region that's circled is the portion of similarity that we found with HIV. So we asked, what perhaps is the structure and what might be these transmembrane spans contributing to? And so we looked at a neural network-based algorithm. It's called AlphaFold2. It predicts the 3-dimensional structure of proteins with very high accuracy, over 95% accuracy. So you don't need to crystallize proteins anymore. You just run them through Google's AlphaFold2. So we did that, and it wasn't able to resolve the structure of the cytoplasmic tail or the aminoterminus, but we had excellent model agreement in the transmembrane spans, shown here for the 3 different models. And if you look at it end on, or this would be looking from inside the cell out, the transmembrane spans actually form sort of semicircle configuration. Now it is the latent membrane protein too, and the potassium transport that we see is in the lytic phase, and we're thinking that it may have a consequence on virus release. So what's known about LNP1 in the lytic phase? Not much. It's unclear what its functions are in the lytic phase. So we looked at sorted latent and lytic cells to see if it's expressed, and we actually find that it's expressed at 63 kilodaltons, which is full length in the lytic cells. And luckily, in Burkitt lymphoma cells, because there's also a MYC translocation, they don't actually need LNP1 expressed in latency, whereas other types of EBV transform cells do. So we're actually able to study this using Burkitt lymphoma cells quite readily using the production system. So we did the same thing looking for localization by immunofluorescence, and we see that at 24 hours, it's got this perinuclear localization here, and then later at the time of virus release, it's relocalized to the plasma membrane. And so with LNP1 localized at the plasma membrane, expressed in the lytic stage, Burkitt lymphoma cells, we turned to our colleague at Ohio State University, this is Harpreet Singh, who's a channel expert, and we asked if he could help us demonstrate by electrophysiology, patch clamp analysis, if it's got channel activity. So we transpected it into HEC293 cells. These are human embryonic kidney cells. And it actually shows a conductance of potassium at 105 picosiemens, and it's specific for potassium but not for sodium, so it seems to be a specific channel. And this occurs at negative membrane potentials, which means that it's a voltage-gated ion channel. So the challenge that we have is that all the functions are known for LNP1 are in signaling via this cytoplasmic tail. So if we remove the cytoplasmic tail, we would, and if LNP1 is just turning on the gene or a host cellular ion channel, we would expect to lose the channel. So we tried that. So we removed that tail, and instead of losing channel activity, we augmented it. So it more than doubled from 105 to 250 picosiemens, and the channel appeared open in both negative and positive membrane potentials, which indicates that the voltage gating was being derived from the cytoplasmic tail. So in summary, Epstein-Barr virus Burkitt lymphoma cells demonstrate increased potassium transport as the EBV lytic phase progresses to the stage of virus release. Blocking potassium efflux prevents virus release. Full-length LNP1 is expressed in Burkitt lymphoma cells, and it localizes to the plasma membrane, and LNP1 forms a selective voltage-gated potassium channel, and its regulation is in its cytoplasmic tail. So I'd just like to thank, these are the members of my lab. I'd like to thank, or point out in particular, Evrys Bio, who's a postdoctoral fellow. Tiffany Fry is a graduate student. She just successfully defended her thesis last week. Jennifer Mitchell is a talented undergraduate student. They all contributed to this data. We collaborate extensively on EBV with the Powdery Lab, which is my wife's lab. These are our collaborators, financial support, but most of all, Tim, I want to thank you for taking a chance on me all those years ago and sharing your wisdom and your recipe for discovery. Thank you.
So first, thank you very much, Mike, and I'm very glad I did take a chance on you. And you were a very safe bet. Thanks for the kind comment. A quick 2 questions. One, does the cytoplasmic tail, does this LNP work as a multimer? You mentioned that in the cytoplasmic tail.
Yeah. So no, I didn't mention it. It is known that LNP1 aggregates in lipid rafts. Most potassium channels are formed by multimers. So there's nothing known. So actually, Jensen Jang at NHLBI is a membrane transporter expert. It does cryo-EM, and we're working with him to see what the multi-molecular structure may be. Yes.
When you block potassium export and the virus then fails to accumulate outside the cell, does it accumulate? Does infectious virus accumulate inside the cell?
Most likely, I don't know. We need to look by EM. We also break open the cells and then reinfect to see if we can do an infection study, to see if they're actually infectious versus not getting out. All right, thank you.
Thank you very much.
So now I have the pleasure of introducing Rob Jordan. So I was in Tim's lab in 1988, 89, and Rob was actually in before me. I don't know if you were his first or his first mentee. And I found out that Rob took the same sort of path that I did, started out as a technician. I think just pulling up for details. There we go. So he's a senior program manager for the Bill and Melinda Gates Foundation. Rob got his PhD at Johns Hopkins University and went on his postdoctoral fellowship at Harvard University. He's been in he's a virologist, he's been in antiviral research, he's contributed multiple antiviral compounds, including a new drug for RSV. He's contributed to remdesivir for COVID, as well as a new drug for smallpox or orthopoxis, which may be in monkeypox as well. And so, and the title of this talk is Antiviral Discovery and the Value of Mentorship. Please welcome Jordan.
Well, thank you very much, Michael, for that introduction, and thank you for allowing me to speak. It's a great honor for me to be here today and to share some of my experiences with Tim and how he's impacted my research career. And that's kind of the gist of my talk is going through some of the things I learned from Tim twice. I worked with him as a research technician. I went away to graduate school, came back to work with him as a research professor, and then moved on in my career. But I've learned so many things from Tim. It's a great honor and a great pleasure. So my talk is Antiviral Discovery and the Value of Mentorship. This was one of the first papers in my career. And so when I left Penn State, I had no formal research experience. I had some techniques, tools, and things that I learned as an undergraduate. And I came to Tim's lab, and he took a chance with me, right? And brought me into his lab, and really taught me how to do research. And it's so much fun working with you. And he taught me how to read the literature, taught me how to put the right controls of experiment, and all the things that you need to learn, to become a good scientist. And I just remember, you created such a great atmosphere for doing research. It was so much fun. And I do remember one time, Tim was still working at the bench as an assistant professor, and I had come in from lunch, and apparently there was a sink that was clogged upstairs, and Tim was furiously working at something on the bench, and there was this waterfall. There's water coming from the ceiling, and he was so gross at experimenting. I'm like, Tim, what's going on? But that gives you a feel for his focus and passion, and I think it was a really great experience. So I left Tim's lab, so I left Tim's lab to go to graduate school and to study entomology, biochemistry. Then I did a postdoc back in herpes viruses, because I wanted to apply some of the biochemistry that I learned at cerebral complexes, which was pretty naive at the time. And ultimately moving back to Philadelphia. And when I came back to Philadelphia with my postdoctoral lecture, I hooked up with Tim again. He was telling me about this foundation, that research facility that was going to build in the world. That's the craziest thing I've ever heard. And then a few months later, I met him up here and he showed me the area, that he was doing. And it was like really happy. It's just incredible to me to see that he was bringing this to fruition. And then he encouraged me to come and join. At the time, I was like, oh, this will be interesting. I can change studying hepatitis viruses, which was interesting to me. And so then I came back and joined the foundation. And I have to tell you, some of the most interesting research was done during that time. I worked with Anand and Tim. We had really had a great research environment. This paper here is a collaboration with Pat and Anand and Tim, studying this molecular pathway in pestivirus. And at the time, there was these two biotypes of pestiviruses, one that would kill cells when it replicated, called cytopathic effects, and one that didn't. And we were trying to understand how the virus kills the cells. And this is at a time we first discovered some of the ER kinases. Pat was an expert in that. And so this collaboration was so fruitful, that we're methodically characterizing this pathway. I still think this is one of my favorite papers in my This paper here is review. For some reason, it's like one of the most highly cited papers. And I remember Tim coming down to our office and he's like, we got to write this review. I'm like, I don't want to write this review. It's like, I don't know what I'm talking about. And Tim will never take no for an answer. So suddenly I see a draft of this review on my desk, and it's like I had to fill in the hepatitis C part, and I wrote which part you were doing. So I wrote something in there, and then Tim made it, one voiced it, and we published it. And it just got picked up, and people kept citing it. When I started looking at the statistics, it was like, really? It was a nice review, but it was just funny. But one of the main projects that I had with Tim is the glucosides. And this is what I came to the foundation to help develop. And at the time, these were post-targeted therapies that had been discovered, periodically over the years. First in 1985 by Schlesingers at St. Louis, Washington University, St. Louis, and there they discovered that if you inhibit glycoprocessing in the ER, you can have an impact on virus replication. So understanding that process, we wanted to develop these for hepatitis C viruses, hepatitis B viruses, and they were broadly acting across many virus families. And my interest really was understanding the mechanism. So we were looking at pestivirus, in this case, bovine viral diarrhea virus, which was a model for hepatitis C at the time. And one of the hypotheses was that these compounds would reduce the infectivity of virus particles. And so in the lower right-hand figure, to prove that, we radio-labeled virus with tritiated urine, isolated the virus in presence and absence of the glucosides inhibitors, isolated this virus on equilibrium sedimentation, and then measure the infectivity of those radioactive virus particles. It was a pretty crazy experiment, but it proved the point. It shows that you don't see too much of a reduction in the viral RNA, you see a dramatic reduction in infectivity. So that to me was strong evidence that these glycoside inhibitors were reducing the infectivity of these particles. This was kind of interesting research, and this is one of the more interesting experiments that I did in my career. And I thought it does typify kind of the quality of the science that we were doing at the center and continues to do. So I left the center to join a company called Biopharm. This was shortly after 9/11 incident, and there was a big interest in biodefense. And so I was my first day at the job that my new boss told me, we want to be working with smallpox. Haven't we already eradicated smallpox? Like, isn't that gone zero? But there was an interest in developing antivirals for smallpox. And so, I'm new to the company. It's my first industry position. So we did a high throughput screen under 500,000 compound library using vaccinia virus as a surrogate. And we found a compound that was really potent and had some nascent structure activity relationships, and chemists got all excited and started developing this molecule as a drug. It had very, so it was very potent in terms of inhibiting virus-induced cytopathic effects. So this is, when the virus replicates the built-up cells, that's staining. But in the presence of this compound, we protect all the cells from virus. And it turned out that the compound itself actually didn't inhibit virus replication, but it only inhibited a small fraction of the virus that's produced from a cell. About 1% of the total virus gets secreted from the cell as extracellular virus. So it turns out this is a highly pathogenic form of that virus that causes the cells to die, well, causes the plaques to form, but also causes the systemic disease associated with smallpox. And so we did a number of studies. So the Biopharma ran out of money, and I had some grants that were supporting the work, and we were able to move that project to another company called Egan Technologies in Oregon, where we continued developing this compound. And after many studies and multiple different animal species, we were able to show that it was very effective at inhibiting orthopox virus replication. And at the time, we were trying to get this, approved, and the FDA had created this regulatory path called the animal rule. But I don't think they expected anybody to actually pursue that, because every time we went to look back at the data, they kept changing the bar, saying that we had to go do something else. I finally got tired of that back and forth. I left that company on Gilead Sciences. But the way I look at this things that I learned in Tim's lab, this idea of just trying to keep an open mind, let the data drive where you're going to go. This is something that I learned from you, Tim. And I remember one of my colleagues saying, "Oh, this is not a drug." Why not? It's like just not taking no for an answer, just being creative, letting the data tell you where to go next. I think that's a really important. So at Gilead, a number of projects I was working on. One was this compound called remdesivir. It was initially developed for hepatitis B viruses, but when Gilead purchased Pharmaset to acquire the Symposium, the molecule was no longer developed for that application. And it was kind of hanging out in the research laboratories, and the company was thinking about using it for RSV, but that's the off-market effects. So I decided that I was going to study this molecule outside of Gilead, with some collaborators with CDC. And at the time, I knew it had some broad spectrum activity. And we found that it was active in Ebola. This was during the 2014, 2016 Ebola outbreak. So Gilead decided to develop it for that indication. Just kind of turn the whole ship around in 9 months. We had completed our phase 1 study, and we were able to actually treat Ebola patients. But we have to see a compound. So the turns out that the molecule wasn't as efficacious relative to some of the other therapies for Ebola. But in the background, I was also working on coronaviruses. At the time, that was not just MERS that we were looking at, because that was an endemic coronavirus. But I had that fully acted on. I had already put all the preclinical data for remdesivir coronaviruses. And 2 years later, we have this outbreak, and now we have this molecule that could immediately go into the clinic for SARS-CoV-2. The problem with the molecule, though, is it has to be delivered by IV. It's a prodrug of the monophosphate nucleotide analog, which is metabolized inside the cell to the active form. If you administer the compound orally, it goes right to the liver. That's fine for hepatitis C virus. And when we develop it for Ebola, an IV route is a fine route for Ebola patients. But for coronavirus, it's a problem. So I think future work needs to try to solve that problem and make it a world model. So I left Gilead. I worked with a few other companies, but my passion for research, sparked by Tim, led me to the Gates Foundation. We're now building a portfolio of programs to tackle the next pandemic. So we're trying to develop antiviral drugs for a number of virus families that we think could be problematic in the future. And we've launched an initiative called Pandemic Antiviral Discovery Initiative, and it's a collaboration with the Evrys Foundation that is trying to fund discovery and development of antivirals over the next 10 years. So what have I learned from today? First off, the joy of discovery. I came into 10th lab not really knowing anything. Tim took me under his wing. He taught me that science was just something that was really fun. I really appreciate that. Grace and creativity that I'm always taking to try to keeping an open mind, thinking about things in different ways. Really, it was exciting talking to Tim about science. I learned so many really interesting things, and Tim came at it from so many different places. It's always a challenge and a treat for me to talk science. Still is. Research is not just about discovering and collecting data. It's really personal. It's a personal thing. Right? I think Tim embraced research with passion that I certainly enjoy and try to keep always, in my career, everything that I want to do, everything that I do is I try to do with passion and invest myself into it. It's really learning from music. Focused on making therapies for patients, keeping the patient in mind. I know, Tim has always been patient focused, especially with the Hepatitis B Foundation and his research and hepatitis work. That was really highly motivating for me. Really connected all the dots. So not just doing research and focusing on amino acid and active site, but thinking about how that's going to be translated to help patients. And that's something I've carried through my entire career. Also having fun at work, and why it's good to have this fun and have a creative environment. I always thought that you were able to create this environment that was a safe place for ideas, right? And I've always tried to do that in all the labs that I've run. Sometimes more accepted than others. But the people who work in my group always liked it. I think that's a good way to do research. And then, being humble, flexibility, being gracious, these are things that I really appreciate about you as a researcher and as a leader and friends. So thank you very much.
One, can I have $20 million? Another timid has always been this idea of broad spectrum antiviral, something that you can use when you have no clue. The pharma never liked that. And I know Tim was an advocate for staff, but no one ever seemed to like it. That changed though?
So I think the COVID outbreak has changed a lot in terms of various therapies, not just antivirals, but host-directed targets for treating symptoms of disease. I think that's been more accepted. Also, the innate immune activator has never been very effective in therapy. So there's been some really promising work through Anand. So I think you're going to see a lot more of those types of therapy studied. And the pandemic, for pandemic, the target product profile is very different than treatment. We want to have something that's probably most exposure prophylaxis, preventing household transmission or community transmission if possible. So it's a different target product profile. So different types of things, sort of things that might be better as prophylactic than treatments. So yeah, I think there's been some changes. Pharma may not have embraced it, but it's from a public health perspective.
I have a question back here, Ron. Enjoyed your talk. So it's a question about the mechanism, about the glycosidase inhibitor. How does it function? Does it prevent packaging, egress of the virus? What on earth does it do?
So at least in the pestivirus case, we always hypothesized that there is local misfolding of glycoproteins. It's getting processed in the ER, and as the virus assembled, it went through the ER into the Golgi, you would have these partially misfolded proteins on the virus surface that weren't as effective at entry. But I'm sure there are multiple mechanisms depending on what the glycoproteins are, what part of the virus life cycle.
Hey, well,
the monkeypox is a new threat on top of the COVID pandemic. So I'm glad you have the ST compound that can treat it, but the virus is evolving, right? So have you identified any like system mutants for the virus?
So we haven't, so drug resistance is always a problem. There hasn't been enough patients treated with these compounds to know what that liability will be. Yeah, you're right, the monkeypox is an interesting phenomenon. To me, it sounds like it's now in the rodent reservoirs. It's not around the rodents. So we'll watch that. It doesn't seem to be as big a threat as smallpox. So I guess the rodent transmission still is not quite understood.
Rob, what is it about remdesivir that allows it to be somewhat broad spectrum across multiple quite different RNA viruses?
Yeah, we really tried to understand that at the molecular level. We actually published a paper. We looked at the structures of polymerases where the compound was active and where it wasn't. It came down to like 2 alpha helices that were in the active. So it just wasn't very satisfying. So we don't know, I think, really, what the selectivity allows it to virus. But it's not all viruses, and so yeah, good question.
Okay, oh, what other question?
Just on behalf of Tim and the Hepatitis B Foundation, would you maybe get the Beast Foundation to
I'm sure I can give it a try.
John?
So I have a non-scientific comment. One of the nicest memories I have from is Tim and I hosted our first lab party, and we were living in Philadelphia, and I remember Tim's idea of a party was to buy a case of beer and like 5 bags of chips, and he invited everyone. The only person who showed up was Rob. And Rob, after half an hour, said, "Is anyone else coming?" He stayed with us for about 90 minutes. He was gracious, humorous, and so we really learned a lot last week from Tim. So thank you for coming to our first lab party.
I'd like to introduce Dr. Anand Mehta. He's a senior associate dean for research. He's SmartState Endowed chair at Proteomic Biomarkers Department of Cell and Molecular Pharmacology at Medical University in South Carolina. So Anand is, when I came back to the foundation, Anand and I shared an office and a laboratory, which was a great experience for me. Anand's such a great colleague and strong perspective, great scientists. And I learned a tremendous amount just by hanging out in the office. I do want to relate one story. It's always in my mind. This is, we were in the office and we were talking about something to do with hepatitis B virus, something new that came up from literature. And Raymond directors the foundation come to walk in. He sits down and he goes, "Well, tell me about this." And it's like, "How did you know that we were doing that?" You know, and then he looks down like, "I saw the trash. I saw that paper." So on it.
But Anand is the SmartState professor at South Medical University of South Carolina. In South Carolina, they have to designate to tell you who's smart. As opposed to the other professors who, well, sorry.
And maybe doing both of these at once is too much for me. But I will say, what allowed me to answer questions when Raymond would come in and ask questions was actually not the Tim, but the Peter blog. So when I traveled around England, in the backseat of their car, Peter would ask questions about the battle between a tiger and a lion, who would win, a battle between a snake and this would win. And I thought, well, I'm here with my boss, I have to answer these questions. So like 3 hours straight, I would answer these questions. So it was perfect training. So when Raymond would come in, I could pretty much deal with him easily. So I've known Tim now for many, many years. And in some ways, I came to this because, one, a retirement party for Tim, I don't believe it. You know it's not true. I know it's not true. So, I wanted to see it. but I spent my time with Tim. That's really spanned, again, the same picture from Jefferson, Google, thank you. Our time, when Tim was here in the Ryan Corner building in Oxford, and I ended up going to as well, our return back to Thomas Jefferson University, I was trying to get a picture of the Delaware Valley College building. I went on the campus, they called the cops, so I'm here. But our eventual feeling here and then Drexel, and then my eventual move, of course, to the south. But I'll kind of take you back in time, and I started in 1992, so I was basically hired in lieu of purchasing a centrifuge. So there were two technicians, another guy who was a student, John Machimolda, a technician, John Messias, and the infamous Doc Finan. So Doc Finan was the student that just graduated, who they were all afraid of, because she was strict and demanding, and she ruled the group. And they were John Messias, who was a football player at Lehigh, got into medical school really because of Tim. Was going to go to medical school in September, and they had a choice. Do they buy a centrifuge, or do they hire someone else to take that place? And so they actually hired me, and about 6 months into it, I think they regretted it, but it was a good deal. But so I started in 1992, and for those that are too young to remember, that really was the start of a great American expansion. I mean, literally, we were coming off, 7% unemployment, a war, around Contra, a whole bunch of things. NIH spending was 10 billion, Alon to 20%. The phosphorylation was basically awarded the Nobel Prize. Tim liked books, the top bestsellers, and I found this to be shocking and disappointing. Give War a Chance by P.J. O'Rourke and Rush Limbaugh are the two best-selling books of 1992. So nothing has changed. But this also came out in 1992. This was the first publication from a group of scientists warming the world with global warming. And it was just something that was done because of the panic they saw. And the first name you kind of see when we always see it is Barry's name, kind of stuck right there, but it includes a lot of people. And it always kind of means something to me when I saw that. So when I started Tim's lab, I was hired. I come for one interview, so there's a theme here. People get hired by Tim. Tim takes a chance on people. That's a theme that you'll see over and over again. I probably would not have gotten into graduate school with that Tim, and not maybe someone else wouldn't have either. He takes chances on people. That's something to learn from, and I would say that to everybody. Why I was hired, I don't know. He thinks I said something smart. I probably didn't. I was probably confused, but I went with it. But I remember one of the first days, Tim gave me this and listed maybe 15 or 20 mutants we were going to make. And I still have this. I don't know which ones we made, but we made a bunch of them. So, it was at the time for me daunting, but something that we kind of worked with. This was actually done after Tim came back. This is an experiment I did Tim with him. When Tim came back from Oxford, we'll kind of go through his timeline. His timeline is in some ways my timeline. We made restaurants together, which was really that Tim proposed the experiments, did transfection, and then I did the rest. But that's how it works. And so it was one of these things. But I did lots of dot with Tim. We made lots of papers. Lots of mutants. We published a good number of papers with both John Messias and John Calder. But he was very generous in publication, and we got a good number of papers out. We also made an Alpha-47 beta gal mutant. Again, because we thought the Alpha-47 gene was not important. It turns out in the Cell paper to control MHC class 1 presentation of herpes virus. So again, we do something. But we made it with a LOX-3 system as well, which everybody uses LOX-3 for mutations now, but in 1992, I don't think a lot of people were doing that. And that may have been Tom, your fault, but we tried it, and it was a mess for herpes virus because of the murder repeats were really doing that. So very early after that, though, after I joined the lab, Tim left. He went to sabbatical. He went to sabbatical at Balliol College. And so he left the lab and spent a year after, and maybe not even a whole year. but I think it was transformational year and changed a lot of people's lives, which is amazing when you think about that year. I'm not going to say how old he is, but it's just a fraction of what his life is, but it changed. And we heard from Raymond. I also talked to Fran Platt. So Fran Platt was a scientist at Oxford, was one of the smartest people I've ever met in my life. Tim Lee, she's now head of pharmacology. But she wrote this very nice letter, and I'll read it out if you guys can read it too. It said, it's hard to know where to begin to describe the fun we had with Tim and Joan. When Tim came on sabbatical as a glycobiology student in Oxford. Tim was a fantastic scientist and a delightful person, brightened up our lives enormously. He and Joan lived near the ring road, which is a traffic circle. So that Tim walked in every day, would pick up stones on his way past the bakery and appear in my office to eat them with me for breakfast, talk science. Tim loves to eat and talk science. For that reason, for reasons that escape me, Tim took to using a lady's toilet, and also that the Swiss stove car, and his sweet coat in there, to pick up his toilet off a hook, and had a spare toilet roll on it that he proceeded to drill in the building. So, I am so sorry, Tim. I have so many fond memories of hilarity at the Wilson balls, hilarity over the Western blocks, hilarity over every aspect of our day-to-day lives. Laughter and Tim go hand in hand. When Tim and Joan went back to the USA, it was though the lights had been switched off in the lab, and I've thoroughly enjoyed meeting up again over the years with Tim when Tim and Joan visited or when she was in the USA. And so Tim, as she said, I can't believe he was hiring. I'm very happy to see you for that career. It's a comment. I apologize for that embarrassment, but I think the impact it had. Everybody in Oxford still knows Tim, not for this, but for this. So Tim did actually a lot of good work by himself, like physically doing work. And he came out of Oxford with a very nice high-profile paper that kind of set the tone of a field of antiviral research that has existed for a long time. And so he had shown that that this small immunosugar, deoxyribonucleic inhibited hepatitis B virus and could theoretically be used. And what he hadn't actually discovered yet, but it turns out the way these drugs work is they inhibit interaction with a set of host chaperone proteins called CalNex and calreticulin. So if proteins can't interact with these chaperone proteins that are fold properly, and if a virus envelope protein isn't fold properly, it produces infectivity or prevents that virus from forming. So that was the basis of the antiviral activity, and from that, we set off on a whole bunch of new work. The other thing about Tim that if you remember, he loved using colored backgrounds in slides. So this is my ode to Tim. So we've done some SAR, modified this, made a more potent compound where we were getting, activity in the low micromolar range, but because of high micromolar range. Still not very druggable, sorry, Tim, but useful. And from that, we got another high-profile paper out of Nature Medicine that kind of showed that you could use these compounds in an animal model and inhibit a virus replication. And so, but also show that you could inhibit glycosylation in an animal model. And actually, I'll get to that in a second too. So that was quite a seminal paper, and actually, it's actually second most cited compared to that oncology paper, which is amazing. And from that study, Tim formed a company, initially called IGX Oxford, then called Synergy Pharmaceuticals. And then he had a joint program at the time with old Bob Moriarty and at UIC. And then it was basically the block group, a bunch of us with a group out of Oxford, developed better immunosugars. And we did SAR, we came up with some compounds. I would make the argument none were completely druggable and drug worthy, but they were better. And this led to several publications that came out. And the best one we have, we call S187. As Rob actually showed, this drug still exists and is being used every day now. They just rename things nowadays. So this is the modern life where we call methoxyminyl deoxynitrogenase is now called MonTMJ. And that's the same thing we developed and is now basically being brought back by a ton of people. They call it UB4. It shows it was dengue virus. And they showed that you actually combine it with interferon and had antiviral activity, which is the same paper Rob published with Tim and he published in 2002. So it just kind of goes with it, things come around. I've actually had people contact me, Tim, to show you that they found that this drug, they knocked out a gene that inhibits all viruses. And I find out the gene is glucose-1. I'm thinking, I'm just going back in time. Which is kind of interesting. But I'll point out the things that Tim's creating, they have a life long beyond what he did. And they're stupid. So then, at a certain period of time, a bunch of us moved to Glycoproteomics. Tim had brought in a guy named Taj Mattoo, who was a student with me at Oxford, and then Taj had hired Marianne coming out. So Marianne, and so I didn't do any proteomic work, zilch. And so these guys kind of helped it up. We wrote grants together, and Taj ended up going back to England and I got the grant. And so we did some seminal work, and a lot of it stemmed from the stuff we did in the animals with Tim. So Tim had shown in his animal study that he could dose an animal and see a change in the population by just doing total serum glycan analysis, which looked at the sugars attached to protein in serum. No one had ever had done that before, ever. So that was the first example of anybody doing something that in some ways seems stupid. Everybody wanted to look at a purified protein. Tim was like, oh, that'd be easy, just do it all. And actually we could see a change. And that change correlated with antiviral activity. We thought, hey, we can see such a small change from a drug. Can we see a small change with cancer developers? And the answer to that, and we did this study with woodchucks and blood tenant. I show a picture of blood tenant. And so we did that study, and the answer was yes. And actually, Tim and I were in Ithaca when the study was unblinded. We were on the phone with Marianne, and we went patient by patient and showed that it was correlated with cancer. And we developed what we call glycoproteomics, where we did glycan analysis, identified proteins that change, ran 2D gels, and nobody runs 2D gels at Marianne, and nobody does it anymore. And then identified proteins that change, and developed assays for that. And this has actually led the companies spinning out, not developing it fast enough, but spinning out, and creating glycoproteomics. This is paper number 4 for glycoproteomics. We developed glycoproteomics. We never talk about it. We don't sing it as much as we should, but it's number 4. And number 2, if you consider the ones that are actually Japanese that we couldn't have gotten. So one of those is GP73. I will talk about that. But this is a protein that again led to another good hyper publication. I think it's the last Tim first author publication. But GP73, as it exists now, is now, there are over 334 results. Again, renamed it. So it's GP73, GALM1, GAL2, and just GP73 with HCC. They're over every day. There's publications coming out, basically confirming the things that we saw and identifying what it does. But what it does, though, it just came out this year, by the way. This is for Tim. It's a hormone. Tim thought it was a hormone. Tim was like convinced that it actually does something. And so, and bloody hell, he was right. So that's the other thing with Tim. He's got some wacky, crazy ideas, but they tend to be right. So what I would always say, if you believe in Tim and Tim method, think a little outside of the box, think crazy, and follow it. You may be right. And if we've done this, we could have had made sure the metabolism. It's actually a good paper. I've actually, like Tim, I haven't read it, but I've seen the data. So post Drexel, we had a split. So in 2015, Drexel kind of called us back. I had gotten tenure, and I thought tenure meant something, so I went down to Drexel, and Tim went road and laughed at tenure. So I went to the new college building, which Tim way is not very new, in Tim's name here. And I eventually moved down to South Carolina. Now, I would say that my relationship with Tim hasn't really, I would say it's not ended in any way, shape, or form. I went to Medical University of South Carolina because, well, first of all, most people have not even heard of it. They think Medical University of South Carolina, what's that? Like, that's some sort of baccalaureate school. But I knew about it because Tim had talked about it. So Tim had talked to these stories, and I don't know if they were real or not, maybe I got confused, but he had once gone through Charleston and the Medical University of South Carolina and found it a wonderful, charming, lovely place. And, I always thought about going back there. So I kind of knew about it. So when I was offered a position there, and then a down chair, I thought, you know, if Tim's good enough for Tim, it's good enough for me. And so that's a major reason why I consider it down. When I did this job, yeah. And so we now become a spatial omics lab. And so the one benefit of moving is you get startup packages and buy a big fancy equipment. So we're a focus of excellence. And so we do basically spatial omics. Spatial omics is just where in a piece of tissue is something. So if you look at, you know, a slide that contains tissue, where are the changes in terms of where the cancer is? Where's your small molecule? Where are a T cell, a B cell? You know, so spatial omics. So we're sort of developers of spatial omics. And so I'm still a glycobiologist by training, and we do a lot of that. So we do cancer biology as well. And so again, example of spatial omics is something like this, where you have different subtypes of hepatocellular carcinoma, which is the main type of cancer that patients with HPV get, where you see that one type of cancer, these highly aggressive X1s, have glycan changes that kind of shoot up bright red right within the tumor. Other cancers, these are the S2 types, they have glycan changes outside of the tumor, field effect type of cancer. And then these S3 type of cancers have very small glycan changes outside, and with a change that's not detected by any marker. What's actually kind of interesting, it came out of the Princeton workshop from Amit and Egan, was the fact that these actually have good outcome post-resection. These actually have bad outcomes post-resection. And that's we postulate because a lot of the field effect that we're seeing here, so we're trying to pursue that now. And these guys actually have no outcome. They actually have bad outcome because they're never caught. These are the mysterious, as you remember, people that are negative on all the biomarkers are left alone. So we try to develop that. So we also do a lot of spatial imaging for lymphocyte populations. So I initially wanted to do a hepatitis B study to see what are the true lymphocytes and immune cells in chronic HPV and reactivating HPV. And I talked to Tim about getting samples, and he was kind of helped us. And then the paper came out with Tim basically for giving that paper. And so we thought, okay, we're switching. So we switched to doing this in NASH. So I have an MD PhD doing that now. But he didn't give me the figure I wanted, but it actually is quite nice. But we do see very distinct differences in CD4s and CD8s in NASH patients compared to HPV. HPV has something interesting as well, and didn't give you the right data. Basically, we see lots of infected hepatocytes that are core positive with HPV that have 10 to hundreds of CD8s around them. But then there are huge swaths of infected hepatocytes that have nothing around, that are completely, they're not being looked at. So by the immunological systems, that remains a mystery. It's something we can go back to if we can get slides 20 minutes out. We also extended that further to say, look, we can do spatial omics on tissue slides and microbiome and microarrays. So we're developing this as a whole new diagnostic platform for analysis for Lytic. Find out another company to do that. So this is just imaging back and into that. So now I'm kind of going to end here, but just kind of talk about what I'm doing now. So I'm now the senior associate dean of research in pathology medicine. And when I took this job, so hopefully no one here is like got a role like this, but this is a loser job. So and I know what I mean by that is this: when you think about like senior associate dean or dean, they're the loser position. They're the positions of people that lost all their funding, that are kind of moving up as a failure. And that I'll say wasn't me. I started very well funded productive lab. I like MUSC. I like MUSC a lot. And I like so one thing that Tim taught me here, it's not about the infrastructure, the buildings, or the air conditioning. It's the people. He loves this place. Tim loves you guys. I can't even tell you how many fights he had with Drexel leadership about letting people go or not because of 2% funding or whatever. He would fight tooth and nail to keep people here because he loves people here. That's why he does what he does. And so that's something I love. And that's why I took this position at USC. I like the people there. We're a small university. We're highly productive. We've got over $400 million a year of productive funding. We have a lot of very productive people. We're small, but I like it there. I could not have done this job or even taken this position if I didn't have this interaction with Tim. The involvement that I had with him was at this building scientifically has allowed me to kind of understand all these people and do kind of what I do there. And in many ways, I do try to emulate the way Tim was here. I've known Tim, as somebody pointed out, for 30, so a long time. I can't even back. So it's a long time. So, you know, a lot of what I do, I emulate Tim. You probably see some comedic So that's kind of what I've learned. But there are a lot of things about Tim that I think, you know, take a lot more time than 20 minutes. But people is one of them. He believes in people. And that's something that we can all learn from. Even the people that don't have the 4.0 GPAs, if you believe them and give them a chance and nurture them the right way, they'll develop and do good things. So with that, I think I'll stop. These are just some of the things in the lab. And send kids on trips. Students love going on trips.
I like your chart.
Yeah.
How about one question?
Very nice talk. My question about, have you rated the super chain to the genomic mutations in the liver sample?
So not specifically, but in general, yes. So we know there are certain sugar changes, for example, that happen we see that happen that are associated with P53 mutations. There's certain sugar changes we see that happen with certain mutations. So there are certain mutations or certain sugar changes that happen with beta link activation. So in that regard, yes, but not in the extreme fine measurement, but in general, yes. Okay, so with that, I'm going to move on. So I went to England in 1995, and I think roughly that time, my introduction will be a little shorter. So it was when Tim Sue joined the lab. So she joined the lab from South Alabama, where she had worked with herpes viruses, but herpes virus is not in herpes virus latency, but in the cornea model. And so the cornea model of herpes virus is what we used in Tim's lab. So she joined the lab to kind of bring that, then also was going to work specifically on the PC12 cells. So like I think a lot of the people that came into Tim's lab, she came in it from the herpes virus angle and then moved to viral hepatitis. And so that's the other theme you'll see. Convert everybody to hepatitis work. And so that was a thing. So she kind of, I don't know if she took over the Doc Finan role, but she was something, she had high standards that we all had to live up to. So that was an important part. So with that, I'm just going to pass it on to her.
Well, thank you, Anand, for the introduction. And yeah, I mean, I remember when I came in, and that's the time Anand was not there. And he came back because I couldn't do encyclopedia, so I learned from her as soon as he came back from summer. And thank you, the organizer, for giving me this opportunity to really stay here to celebrate Tim's retirement. And this is really a great honor to me to really give a talk for, from the herpes symptoms. Okay, from herpes symptoms virus research in hepatitis B. And that is 27 years journey of the discovery and his mentorship. To put the 27 years in a 20 minutes talk, that's a little bit difficult, but we see. That's 27 years ago. That's 1995, and it's about a couple of years after Tim established research in HBV for the reason you all know. And I came in to pick up the herpes symptom project and try to understand why herpes established latency. And there was in the herpes field, that was my mentor told me the area you don't want to get is too crowded. And so we focus on polio infection, but here comes like into this very, very crowded area. And try to understand why virus is their latency and the activation. And then as a lowly postdoc, learning how to run excited PCR from Anand, and how to grow the PC12 cell and differentiate into the neuronal-like cell, or to use as a tissue culture model to study herpes simplex virus latency for about 10 years. So one day, team came back and very, very excited and told me, he found somebody in the flight, a stranger, okay, and this guy told him, you can detect colon cancer DNA in urine. And Tim said, no, I don't think. Could you believe that? That's hard to believe. Well, if this guy can do it, we can do it. So very quickly, he put a $100,000 grant together in 1 week and get us funded and introduced me to the cancer research and funded by NCI. Actually, I was become the first batch of the associate member for the ERN. And then we got the hundreds urine sample from UMich. And yes, we did. Okay, he's right. It's a lot of people say he's right. Okay, so although it sounds really strange, but that's what happened. So we published a paper showing the human urine does contain DNA from blood, but the person has a cancer. For example, in this case, colon cancer. You can get colon cancer DNA in urine. And as time goes on, we show the liver cancer DNA we can also detect in urine. But how about the virus that infect liver? Yeah, we can detect also fragmented HBV DNA in urine. So if you look at this slide, was pretty complex, but I think most of you kind of probably will have to agree with me. This is probably just a very small snap of what is in Tim's mind. So sometimes, I wonder, okay, the difference between these two men is probably just hairstyle, right? Okay, so before I really, so this, I like to share, with 27 years working with Tim, probably is the longest his colleague in research. Anand know him for more than 30 years, but Anand in and out, it doesn't count, okay? So I'm staying with Tim for 27 years. I even lost the count. I sent the slide, correct the slide with Megan. I said, come on, if you can change, because I lost 10 years. 10 years is like nothing, right? 27, okay. So I think I kind of know the secret of Tim's success. Of course, Tim's great mind and his sincerity and hospitality. There was in Jefferson as a loading postdoc. I come in every morning, and the first question from Tim is, "Oh, do you want a cup of coffee?" And then he will just get and give me a pour coffee and give to me. And then it comes to a second question. Is there anything new? I don't like this question 3 times a day. That is very annoying. Even my kids will ask me, is there anything new today? I said, enough is enough. 3 times, that's enough. We don't ask for that anymore. So he stopped for once a day after I come back. Okay, so, but a cup of coffee to a lowly postdoc, that's pretty much it's like a culture shock for someone as a Chinese. Okay, it's just but I really don't believe just a cup of coffee in the office can make such a great success, right? I think so. But I think it's really the bed and breakfast in and countless dinner in his house and even months-long Airbnb that John runs. Okay, so we say behind every great man, there is a great woman, and you know who she is. And I was shocked. I was someone's wife. Tim told me one day, he said, "Oh, I met someone and sit next to me in the train station and in the train, and we had such a great conversation. But, oh, the train is going to end, so I don't think it's enough. So I just invite him to my house for dinner. And all I need to do is call John. And here comes the guest for dinner. And I say, what? I mean, how, I don't know who else can do that. And John seems very used to this spontaneous pop-up invitation for their dinner. And I think a lot of ideas, a lot of collaboration really come out from his kitchen table and dinner table. It's not just a dinner, okay? Sometimes it's a stay overnight. And I don't know how many people has been hosted in Tim's and John's house for over months, okay? And everything provided, breakfast, dinner, and snacks, I guess. So I don't know how many, but at least I know these three. And that's the first one is Emily Thompson, who came to learn, is Barry Blumberg's granddaughter. So she came to learn the virus that his grandpa got Nobel Prize for. And she lived in his house for, I guess, 2 months. And she's from New York. But the guests come as far as Africa, from Ethiopia, and Jonas lived there for a couple months as well. And Maria moved in because the center had a power outage. Okay, so that's, and with this, I believe that's how Tim, I'm not saying he's not smart enough to do research, but with his hospitality and with John's really like unthinkable support, seriously, as I say, I'm someone's wife, I don't think I can handle that. To really support. And their hospitality really build a really nice network, and that's how the foundation started, and Blumberg and Biotech Center. Let's see. Okay, so, and also a lot of fun discovery research other than just the institute. So what I like to do is really give a switch to the research. Okay, that's why I do it. Okay, so just a very brief introduction for herpes simplex virus. This is as a team's postdoc, I then try to figure out why the herpes infection causes a latency, and this latent infected virus got reactivated. For the most of human, the herpes infection in human is most asymptomatic by reactivate and cause a fever blister by spontaneous, mentioned young people, and then poliovirus is frequent reactivation. All the encephalitis is variable to the brain. So if we can somehow stop this reactivation, maybe we can prevent all this. But the problem is, when you try to look at the latency side, which most of them in trigeminal ganglion, try to look at, I'm so thankful we have McIntosh and Robert give a very nice introduction about herpes. And the only thing in this latent infected neuron is latency associated transcript. Okay, so, and this is the intron. Okay, for RNA people, for DNA people, you know intron should be very unstable. So why you got this could be up to 1,000 copy of intron in this infected neuron. And you all know Tim can talk, and he likes to talk, and he talked everywhere. No, he was the time, there was a time he joined the hepatitis research group in Berkshire, and he came back one day and said, I think I know why this thing accumulated, because I talked to John Taylor, who is a Delta virus leader, and Delta virus is RNA virus. He said, I bet it's that. If it's not circle, it's variant because it's intro. So let's take a look. So what do I know as a postdoc? I listened to him and went to Nigel's lab and killed a lot of mouse and bring it back. And set up a collaboration with John Taylor. And then we take a look for, well, it's the intron, so it's really the intermediate of RNA maturation, the process. So theoretically, most of the intron is going to get debranched, and then just disappear. RNA is linear, it's very, very unstable. So we said, well, something may be wrong with this debranching. Okay, so when you look at when you compare the consent branch point in simplex virus, is somehow not consensus sequence. It's only 3 out of 7 nucleotides conserved. So if that's a possibility, maybe the branching itself is very inefficient. So if we make a mutation, then it should be gone. So here comes, yes, when we start to do in the plasmid, and once we change the non-consensus branching sequence from wild type to consent, to the consensus sequence, then the RNA is found. This is the that transcript by Northern blot organization in tissue culture by transfection. And at the same time, we also make this mutate. Well, if you're not sure, you mutate, right? You find the answer. That's how you mutate. So you mutate more sequence and put in the virus itself and looking at the infected neuron. So if it's a wild type, you see so many variants with the stable accumulate in the neuron. But if you change to the consensus branch sequence, here comes, this is the northern blot from tridemic area of the mouse. And with wild type, you see 2 heavy lag intron accumulate. But once you change, you make mutant, a change to conserve, then you don't see it anymore. But you make a rescue back, and here comes state. So actually, this is really the discovery of the first very stable intron in nature. Okay, you can make artificial because you can do cytoreductogenesis, but this is really the discovery of the first case, the stable nature intron. So let's switch gear a little bit. Since I'm working from her to simplex to hepatitis. As I say, we found with the team's great idea, something he didn't think it happened. Okay, it's possible, but and we also showing that we can detect liver cancer DNA in urine. So can we detect liver cancer DNA in urine for early detection of liver cancer? Yes, we have a test now that's 30% more sensitive than AFP alone. But when we look at this, as you know, the DNA, if you look at TP53 mutation or other, you don't know that's coming from blood or coming from liver. So what can be a liver cancer specific DNA? And how about take advantage of HBV, HCC that has HBV DNA and if you detect in urine? I guess that's a pretty. This is sample patient still, okay?
Oh.
Just few weeks ago, okay? And I mean, we argue a lot, we disagree to each other a lot, and the favorite word is maybe we discuss a lot, okay? So, whose idea was it for using viral host junction DNA sequences as signature for the infected hepatocyte to monitor the clonal expansion? And that conversation was happened a few weeks ago. Still, he thinks that's his idea, and I think that's my idea. And well, at least I get to test the idea. Okay, so I get to test. Okay, so this is really the model how we the origin hypothesis here. Because the HBV infection, HBV DNA can integrate with the host chromosome, but the integration site is random. So basically, what this means, if every integration will create every new unique junction. So for the liver, there's no viral infection. We only look at urine, and you should not see any HBV DNA. But for the virus, there has a lot of integration, and you're going to see a lot of the different junctions. But you know the cancer is an uncontrolled clonal expansion. So once the virus mark the infected hepatocyte. If this unique cell growing to millions cancer cell, if you look at urine, that's probably the most abundant integrated site. So to test this working hypothesis, what we did is we collect this DNA from urine and run the next generation sequence and identify this most abundant junction sequence and develop a specific junction specific PCR to see if this sequence we identify is really coming from liver tumor. And in this case, that's let's see if I have that here. Okay, so yeah, so this is same sequence. I don't know why this is going wrong. Okay, so but anyway, out of the 10% of the junction sequence we examine, yes, but somehow we discover this chromosome rearrangement. Okay, this is the example. We find the sequence, we develop junction PCR, and we try to PCR out this region from tumor and from urine. And urine is longer, that doesn't make sense, right? So we say, well, and then we run the sequence, sample sequence, and we see, well, there's 23 base pair insertion. So what we did is design another primer and try to see if this original sequence, the first junction was there. And here comes it there. So, so we detect not just viral host junction, and we also detect the chromosome rearrangement. And so I guess I show you the junction can be detected and as a potential biomarker for tumor or uncontrolled total expansion, although we haven't settled for whose idea this is. And we also detect the cancer-derived chromosome rearrangement in urine. That I think is a potential for cancer genetics. I just like to show one of the example we can use this urine, and not with skin, with this kilogram. Okay, so this is work that Peter, when he was doing his residency in the Jefferson for internal medicine and working research with Dr. Han. And we happen to have a 800-ish urine. So they start to, and we look at, so again, we detect the junction from urine and we monitor this specific junction with data for the recurrence of the liver cancer, okay, for the liver cancer recurrence. And the bottom line is you can really detect this recurrence of this junction even before the MRI can tell you this person has a cancer recurrence. So I'd like to end up my talk with the spectrum of Tim's mentorship. Okay, his mentees, 20s, and even long back. A lot of you might not know he mentored someone at this young age, and that's also my kids. And Tim was their mentor and nominate them to get this $25,000 scholarship from Davidson Foundation, both of them. And this email tell you how that started. That time, Jenna, my daughter, was in high school program, and John was very nice to drop a Davidson Foundation information to my mailbox. So I thank her for that because I never heard about this foundation. And turned out it's because Peter said, well, I Jenna is doing real science, so she might have a chance. So they drop over. So in return, John not just provide the information, provide team to and volunteer team help. Okay, so if Jenna want to apply or help, I mean, team will help anyway. So team end up, support Jenna's application. So they got, so she got award for $25,000 scholarship for this very prestigious award. It's only 15 to 20 young people per year in the area from literature, music, science, and technology. So you know, Tim wrote a very, very strong recommendation. And 3 years later, he wrote one for Ben. Okay, so both of them got that. And 15 years later, the founding scientist become a biologist. And this is a screenshot from my daughter Jenna's when thesis defense in a couple of years ago. And the top panel in his acknowledgement slide, top panel is a thesis committee member, and she specifically, specially thanked him to be his first career mentor since she was ninth grade. And not most of you probably doesn't know that, except Erica and Judy, because they write the letter on their different they start the letter for so many different applications, even from the college application to grad school application. So they know I'm giving this talk. They really want to thank you for that. And I think my whole family, my husband, Frank, is also here to celebrate your retirement. He really retired. He did it. And why? And why here? And I think this tells you the answer. This is the email sent by Kim and to his long-term collaborator, Dr. Han. And I like the subject of the letter. The email is a legacy collection. Kim cannot resist to mentor his grandchildren. Okay, that's okay, so I think that because the Henry, I guess, Freya, so they and Peter is doing his fellow, hepatology fellow at Yale. So congratulations to that, and thank you, Tim and John. Really appreciate so much for the impact on my family and many others. And I think we enjoy your retirement and have a great time with Henry and Frank. Thank you.
Thank you.
Question for Sue? Yes.
About the viral DNA in the urine or the carcinoma. So many of these cancers are preceded by chronic active hepatitis indications. So chronic active hepatitis often leads to development of HCC, but not in everyone, as I understand.
Yes, that's correct.
Is there a way to think to go back and actually start looking earlier in chronic active hepatitis like we did? Or even diagnose these tumors much earlier than standard pathology?
Actually, that's what we are doing now, because integration, you can have a recurrent integrate the oncogene, okay, and you can detect those junctions even when they are in hepatitis or the cirrhosis stage. So if those integrations sustain, they might be up and down like a church. Okay, for people, we have the longitudinal sample from collection from Dr. Han. And for the like a church integration, you see up and down, and that person keeps as a cirrhosis patient. So, but for someone you see it continue, I think that's yeah, yes, that's what we do.
Thank you, Susan.
Yeah. Hi, everyone. Tim, yeah. Here we, congratulations to your achievement, not your retirement. You have new rules, right? So it's my great pleasure to introduce Junhao as my brother. Unrelated. So there's a Chinese saying. So if you have the same last name but unrelated, so you 500 years ago you are same family. So he's my brother. So Junhao now is everybody knows him here. It's a Tom London distinguished professor here and also the VP of research at the Blumberg Institute. And when I was here for about 9 years, and more than 9 years actually, Junhao has been really a great friend and a mentor to me. I learned a lot from him. I'm still learning from him, but he didn't tell me everything now. So now I have Junhao had some comment on the out-of-the-box thinking of Tim. So Junhao.
So, I start my career to study hepatitis B infection. So as a graduate student. So, I joined the institute and that's that was 2004. So before that, I do know Tim from his publication and also from the occasional meeting in the annual hepatitis B meeting. So basically, I think I was actually in a very unique position from the outside observer to the colleague inside collaborator, and over the 18 years we together did lots of the things. So I just want to highlight a few things and what I think about the team has not only done for his personal achievements, and also what he has done generally for the hepatitis B research community. So I'm old enough and like history. So basically, I want to put his career into frame of the history. So when I start work on the hepatitis B, that was just the golden age of the HBV molecular biology. So the HBV, that was actually the first human virus that have a whole genome sequence was get, because it's too small. That was in the 1980s, okay? And then have all the molecular biology and try to understand the viral application in the 80s and expanded to the middle of the 90s. At that time, is every year have a lot of very exciting things appear. So I was when I get into graduate school, my mentor is actually work on antiviral drug discovery. So I just when I have a lecture from the professor from the Oxford University. First time I hear the things that called self-directed mutagenesis. I just feel so excited, and I go back and tell my mentor that no, I'm not going to do the antivirals. I want to do the molecular biology. So then she was so nice and supported me. So then I get there, and then finally I joined the faculties and as initially as a postdoc there. So I meet Tim there, as I believe that was the earliest time is 1995 or 96. I remember clearly that you and Sue, participates that very nervous the biology meeting, right? So at that time, I over the years, I think that Tim is because the Anand have talked about a lot and Sue talked a lot. I from my point of view, that Tim is really out of box thinker, especially in the scientific development. So as listed here, so every decade he seems come up with some very interesting idea. So the first, when he starts the work and just looking for try to cure hepatitis B and try to reduce virus particle production and reduce surface acid. So as a virologist, I think at that point, most people will not take that, because the surface antigen production, that was not an integral part for the HBV replication, right? And also at that time, the target viral polymers with the nuke, that was actually is just approved works for herpes, for HIV, right? And later on, actually 6 or 7 nukes have been developed for HBV and used today. But at that early time, team actually choose a very different way to address these things. And Anand have already shown this. So they try to target the host enzyme to catalyze the unlinked glycan process. So if you inhibit this process, that's the glycan protein will not have a proper interaction with the chaperone and then mostly aggregated. So we use these early compounds that found that the treat with those glycan inhibitors that can actually reduce the intracellular viral envelope protein. So based on this label that was the habit, I think this is must take its own work. Because other people will not label that L protein as S1.
That's not a giveaway to us.
And so that he demonstrates in woodchuck that, again, this is a very typical TIMS presentation. This is the in a percentage of change of the virus DNA in the treated woodchuck blood. So basically, you can see that the dose benefit reduce the viral DNA in the treated woodchuck. But I still don't understand that the control that actually is increased. Right? So anyway, that was clear health efficacy. However, the thing is complicated over the next 10 years in the field that gradually learn that those compounds, treatments that can induce the intracellular degradation of L protein and the M protein, and even the glycosylated S protein, but have no dramatic effects on that unglycosylated form. And this unglycosylated form actually is can be from a subviral particle that's secreted out of the cell. So basically, you can reduce some of the viral particle production, and also due to the glycan change, the produced virus have reduced the infectivity. However, cannot significantly reduce the surface antigen in the blood. So that is actually not partly achieves the goal of the team originally wanted to achieve. Reduce virus loads, but not dramatically reduce surface antigen in the blood. So at this point, that Anand have already told that. So one branch is the glycan study, developing to the diagnostic and also the glycobiology project. Another branch is that it takes advantage that at that time, as I remember, that was around the 2000s, that West Now academics in the US. And then Tim just realized, oh yeah, this can be a broad spectrum antiviral and applied to this emerging infection. And those things, and in the next 15 years, and several people, and then Jian Hong joined the lab, institute, and finally, demonstrates those inhibitor of the group studies can inhibit the dengue. This is all in vivo data, and also can works to protect the deaths by the Marburg and Ebola. And I think that over these 20 years, team have been gets grants funded from this immunosugar project. Roughly, I calculate just close to 30 million. So I think that I have been supported. A lot of my work is supported by this sugar project, and actually, Tim just allowed me to not actually directly work on this sugar project, but work on something else. So my direct contribution is that one of my postdocs at that time, that develop, work on the interferon-induced protein that inhibits virus entry, and work on the human coronavirus at that time. And he developed a very interesting cell fusion assay, and shown that, yes, this compound is very, very interesting that can inhibit the coronavirus. At that time, it is SARS-CoV-1. That's member fusion. And the interesting point is that previously, people just said this compound that inhibits the glycan process of viral protein and inhibits the virus entry. So his work actually found that the compounds treatment also inhibits the glycan process of the ACE2, that's the virus receptor, also the SARS-CoV-2 receptor. So that's by inhibiting the ACE2 glycan process, that's Anand have helped us to identify that the glycan patterns really changed by drug treatments. And you can see that if you inhibit the spike protein glycan process, you inhibit fusion. And if you inhibit the ACE2 glycan process, also inhibit fusion. So basically, that was double hits, both on the virus spike protein and the glycan protein. So this knowledge was actually, in the SARS coronavirus pandemic. The paper is very well cited and demonstrates unlinked glycan in the N20 position of the ACE2 is really important for virus binding to the receptor and triggers member fusion. So that was my direct contribution to this course. And of course, so now that's back to the HBV, and the team's goal have not reduced the surface antigen, have not achieved. And at that time, the Andy Kokonagi joins the institute and initiates the project. And screened the compounds and found this first in class surface antigen secretion unit, right? And later on, the team have collaborated with Emeritus and work on a new class of compounds we generally called DPhil. That is the inhibitor of the host, non-canonic RNA polymerase inhibitor. So these compounds that can selectively reduce virus RNA, and how that works. So Telonjo and team their work is the first time shown that the viral RNA, there is called PRE elements, this stem loop. This stem loop structure is essential for DHQ to induce virus RNA degradation. Of course, later on, other groups shown that there is a host cellular RNA helices can bind into this stem loop and recruit this RNA polymerase to elongate the shrinked RNA poly tail and extends the RNA half-life. So this is, I think, is a very important contribution. So, initially, I really, is not a fan of, try to reduce surface antigen. I think that was just a marker for the cure. But recent years, viral immunology study gradually let me convince them that, the surface antigen is indeed a key to induce the immunotolerance. And you may be essential to reduce it in order to activate the immune response to achieve the functional cure of hepatitis B. So this is a review article I was published last year, to discuss this. And we actually now in my lab have developed a new system, very robust, to looking for the compounds that inhibit the surface antigen secretion and induce cellular degradation. Of course, those studies that was really based on our most recent, this molecular biology study found a key motif that was essential for the subarachnoid body. We believe that we do have a chance to directly type the surface antigen. So next one is also very interesting that, Kim is actually the first one to advocate and thinking we should directly type the CCD. It's not because that everyone is not thinking the CCD is not important. Actually, early on in the 90s, people believe that CCD that is essential for the persistent infection of HBV. But people just think, I don't have a way to, directly target based on the molecular biology understanding. But Kim is really the first one trying to do something on it. I remember that was in the early 2004. So Cohen have joined Tim's group at that time. And one afternoon that he called me, at that time I was still in Christoph's lab in Fort Chase. That's, oh yeah, Kim attended a meeting in Arizona or somewhere. And he said that looks like this CCD is so important. We should do something and work on it. And see if there's a way we can actually do it. Because we have worked on this molecular biology, this circle for many years. And we actually discussed that in our cell line that E-antigen is actually can be a marker to do that. Of course, that's, can is actually immediately act on that. And then when the head is come here, and also, we actually joined the institute. He 1 month ahead of me. So basically 1 week, right? Yeah. So that's can initiate, and also all this data was generated by head. And you can see this is a typical head styles of the figure. Okay. If you look at his paper, that's always this kind of very well lined up and detailed analysis. So long story shorter, so he clearly demonstrates here that was the E-antigen secretion is parallel with the CCD accumulation. So, and Andy and had thought that they work together and use this assay and screening the 80,000 compounds library from our institute and found these 2 first generation of the cccDNA input. And had thought later on, of course, he will tell you that further improve this system that was get a new patent and have been used that for the compound screening and also the basic virology studies. And with these compounds, had have get his first grants and start his independent career. So last one, that's about 10 years ago, that's Tim tell me that, well, yeah, how about we learn what's the CTL, the satellite T cell, do to clear the virus. Select kill the infected cells. We should do something, develop a chemical T cell, right? But I just say, great, but then how can we do it, right? Tell me what's the difference between the infected cells and the non-infected cells? What's the difference we can actually use it? So, but Tim is consistently trying to pursue this project over the years. Several students who have put on there and looking for the company. Of course, it's very complicated this time. And until 2 years ago, and he started to talk about requirements, and until now, that's every few months come up with some new idea, try to do this. I think that this will be continue his up to requirements project. Okay. Yeah. So yeah, this is just for the therapeutic development. You see, he's really a leader of the new therapy search. And for the community, I think that several people have already talked about. He founded the Hepatitis B Foundation, established the Blumberg Institute, and the K-Bot Center. So, especially for me, that the Blumberg Institute is really a platform for my career to develop, and for the years that several people and use Blumberg get his first faculty job and also advance their career. And we once I think that with Anand and I told her, in that few years, I think this world's largest HPV research center, right? You have so many people focused on the HPV research. So finally, I think that he is really the leading KOL in the field. So I just say that the foundation gave us a Blumberg Prize in the over last 20 years to recognize people that have been made a contribution for the hepatitis B research. And previously the HPV annual meeting was initiated by J.C. Summer and Howard Warmers in the 1983. But until 2005 or 6, right? That's every year the faculty needed to fund all the resources and to organize a meeting. Since he became organizer in the 2005 or 6, right? 5. So after that, all the organizers don't need to have the administrative power, and the foundation take over to as anchor of this meeting. So everyone really, recognizes this very unique contribution. So lastly, Princeton Workflow. So this is, the workshop that was very unique. Can gather government officials, industry, drug development leaders, academic scientists, all together to trying to do this small group in big discussion. And initially, that was started from the 95, and this one is 2000, year 2000. The summary of the Princeton workshop. I highlight few. At that time, you think about only drug approved is not pegylated interferon. It's just regular interferon and removed. And you can see the subject as people can just talk that, yeah, something else is required, and also just start, think about the drug resistance, all these things, right? So if you look back over last 2 decades, we have gone very far, right? And this is 2003 one that you can see lots of the familiar face, first here and here, right? So this year, the subject is a cure for chronic hepatitis is really possible. And with that discussion, I think that team is really the first one to define what's a functional cure. That should be the goal for all the new therapy. Okay, and later on, he have gathered both academic scientists and some of the industry leaders. You see, from this research agenda for the cure of the chronic hepatitis B. And this document is not just a hepatology publication that actually have an impact for the policymaker in the NIH, and also, for the advocation that was the DOD to fund HPV research. So actually, I was the first one to get the DOD grant from this as a consequence of the advocacy. So finally, I think that I don't think that the team were really going to retire. We're going to just change the way we're going to work, okay? And that beside what I have right here, that I think, over the last almost 20 years, we work together. We are very pleased to be achieved a lot. And I was especially want to thank him, his confidence on me. And also, we have a lot of, fights, disputes, and, use Tim's words that was not sugarcoating. Okay, so I really appreciate your big heart and really, that your support to my career development. Yeah, thank you. So next is my younger brother. So we have been, colleagues, start from the, 4 kids, and we just at that time we were smoking, and we talking outside of the building, the science, all of those things, and then he actually gets the job here first, right? And then that he talked about come here, and Tom London come from my course my lab. I said, oh yeah, that foundation have opening, because at that time I'm looking for a faculty job. So that is a good opportunity. So I come and I feel that yes, there's going to be an opportunity. So then we almost come together, and here we have been working together for 8-9 years. That was, I think that was the most productive and years in my career we worked together. Yeah, please.
Yeah, Tim left. So I hope he'll stay. But the field needs me. He needs to stay.
You said I'll be back in 20 minutes.
I'll finish that. So currently I'm at Pittsburgh. I left here and then moved to Indiana, and then in 2019 I moved to Pittsburgh, back to Pennsylvania. So I'm going today to talk about HBV loss on the circle. The circle means that covalently close circular DNA, cccDNA. So my time with Tim, briefly, I can come back. Just start. So as Tim mentioned that, Tim actually is among the those early people who call for like elimination of cccDNA as a cure for hepatitis B. So it's like cleanliness, the cccDNA is good. Everybody needs to know that, right? It's not just and that time he called it Bill Mason. See, there is a job opportunity there, so I joined here in 2004 at November, 1 week before people joined here, and so that's at that time I'm a new kid on the block. And then after in the following years, we have been work hard to tackle on the cccDNA biology and try to develop assays to look for cccDNA inhibitors. So working with Tianwen and DPhil, we have established this system using the E antigen as a surrogate marker for cccDNA in cell culture model. Originally it's 8038 cells, and then later on we modify that and improve that into D19, DS19, and then the Hep B HAE82 cells. And using the system, and we have been working on the molecular mechanism of cccDNA biology. And we have first identified, this DNA intermediate called deprotonated HBV RC-DNA as a precursor for cccDNA with DPhil and other scientists here. And this has been really a breakthrough in the cccDNA formation study, and lead to many studies around this cccDNA formation mechanism. And using that E antigen reporter system, we have been to drug discovery for HBV inhibitors that including this chlorophyllid, like, broad spectrum antiviral agents, and also using the CCC DNA inhibitors that people mentioned that design and then in 2014, that marks my 10th year at Doylestown. At that time, I decided to leave to develop my independent lab. Dr. Block didn't block me. And he was very supportive, and I set up my lab in Indiana for 12 years, and I made a lot of money there. Research fund, and then I moved to Pittsburgh. I think Doylestown, I consider this is like my hometown in the United States, and because I scientifically grew up here, and this is a home visit, so I'm very glad to be here today. And that at that time, very productive and rewarding period for me. That's the 30 papers that I co-authored with Tim, and plus 2 patents. Thank you, Tim, for your support. And this is my current research. I put this all because that means I have multiple legs in my research. So I do a lot of things about HBV, not much on immunology, but all other things. And recently, because as a virologist, we all work on SARS-CoV-2. I'm trying to develop a rapid myself for SARS-CoV-2. We published a trend rapid system, but we still don't have a stable rapid yet. And these 2 virus are quite different. Small double-strand DNA and largest single-strand molecules. So now back to the cccDNA business. So everybody knows that as Tim promoted that concept, cccDNA is a central molecule for the HBV leukocyte, and its elimination is required for elimination of hepatitis B infection. Right? But unfortunately, we don't have the drug yet to eliminate cccDNA. The current antibiotics, they are not, or only indirectly target cccDNA. So the cccDNA formation, how it's made, that including a dynamics process, including the transportation of the rcDNA into the nucleus, the encoding of the rcDNA, and then the conversion of the rcDNA into cccDNA. Those are the major questions. And this formation step, a major step, is a repair step, because the rcDNA, you can consider this as a damaged DNA, because it has gap, nick, and terminal modification on that. cccDNA is very clear, circular. And remove the covalently closed polymerase from the rcDNA is a mandatory step in the cccDNA formation. So that's why lead to us to discover this intermediates, which is called the deprotonated rcDNA or protein-free rcDNA. This is the intermediate of cccDNA formation and the precursor. And the work on this DPrcDNA as a central molecule and other people, we have worked out a model for cccDNA formation, starting from the deprotonation of the rcDNA in the cytoplasm, and then it leads to the nuclear capsid structure shift, which exposes the nuclear position signal on the capsid, and then transport the rcDNA into the nucleus. After encoding, the DPrcDNA is repaired into the cccDNA. And the repair process is the essential biology function of the cell. So the virus hijack this function to do this stuff. But compared to the typical substrate for DNA repair, rcDNA is not a typical DNA repair substrate. So that leads to a hypothesis that maybe the cccDNA can be formed by multiple DNA repair factors. So there's a redundant function among the DNA repair machinery. And there's a manual approach to look for those DNA repair factors, including proteomics and other individual chemical assays. This summarizes the current knowledge about cccDNA formation mechanisms. So there are many factors have been identified, in the cccDNA, play a role in the cccDNA formation, that including polymerase, ligase, the endonucleases, and also topoisomerase, and there are DNA sensors like ATRCH1. And also this works from MATLAB and DPhil lab and other people's lab. And this is a very complex process. And recently, we try to identify novel factors involved in bioproteomics approach. Basically, we design, oligo, a biotinylated oligo to target, the single-strand DNA region of the rcDNA in the nucleus, and then pull down the rcDNA protein complex that identify the protein may be involved in cccDNA formation. And that was done by graduate student Alex McIntosh. And we are among the list, there is one protein called DDP1, DNA damage protein 1. That is we prioritized that for the first study. And UVDDB basically is a complex of DDP1, DDP2, and the D3 protein lattice complex recognize DNA damage and then initiate the NER pathway to repair the DNA. And this has been validated by siRNA knockdown assay and also the chip assay showing that both DDP1 and DDP2 bind to the nuclear rcDNA in these cells. And then also in knockdown or knockout DDP1, you can dramatically reduce the cccDNA formation, and also another intermediate called CMRCD. And knockout DDP2 in the infection system also reduce the cccDNA formation and the infection. That's done in NTCP cells and also in the primary and other cells. And then if you express the DDP2 in the knockout cells and rescue the cccDNA formation, that means this is it's not an off-target effect. And if you overexpress, other the functional defective DDP2 mutant, like the ubiquitin deficient mutant and DDP1 binding deficient mutant, we can also rescue that. That means those functions are required for UVDB complex to repair rcDNA into cccDNA. And then due to this updated model, it's actually big. So from the rcDNA of the deprotonation that enter into the nucleus, and the UVDB complex recognize the DPrcDNA. Most likely, our data show that it binding to the manner strand, the overlapping region there, and then initiate the GGNER, the global genome nucleic extension repair pathway to repair cccDNA, because some of the previously identified DNA repair factors involving cccDNA, they also on the NER pathway. So still, we need to know how the rcDNA, the protein is removed from the rcDNA. We still don't know the enzyme yet. And the cccDNA after form is not just a native DNA. Actually, it's a mini constant. It's decorated by histone and non-histone proteins based on the electron microscopy study previously. And the mini chromosome, because of the histone associated with it, undergoes the epigenetic regulation. And there's histone modification, DNA methylation, and other types of the histone the epigenetic modifications. And the HBX protein is a major factor to shape the cccDNA at the epigenetic active stage. And this also shows that the HBX is important, for maintain the cccDNA transcription in the infection system. If you use X matters first, you cannot have transcription, but you still have cccDNA. And this is in the infection system show the epigenetic markers associated with cccDNA. If you don't have X, so the positive marker is reduced and the suppressive marker is increased. And then we use a cell line that's to we knock out the X protein from the transcript and make a cccDNA, but without the X protein, that cccDNA is transcriptally inactive. By using this system, this is just show the epigenetic markers associated with wild type and X matters cccDNA. They are completely opposite. And using this system, we compare the protein associated with the wild type and the X matters cccDNA, try to identify the host factors to regulate the cccDNA transcription, right? So based on the proteomics hit list, you will see the proteins in the green box. That's the proviral factor for cccDNA epigenetics, and this white box, this should be the silencer of cccDNA. And in the overlapping region, that could be the scaffold protein or just the core protein inacting factors, because we use core amyloid to pull down the cccDNA, because the core protein is a part of the cccDNA chromosome. And also, one of the hit in the silencer category is HMGB1. So this is the high mobility group box 1 protein. Actually, it's a very famous protein in immunology field. It's a cellophane, and it's a dam. It's damage associated molecular pattern, and can initiate inflammatory response. And but also HMGB1 in the cell is also a DNA binding protein and it regulates host DNA transcription and repair. So we think that this maybe is one of the factors involved in cccDNA epigenetics, and it is. But you can see the the binding of the HMGB1 to the cccDNA in the X minus condition. That indicating it's probably a silence of cccDNA in the absence of X. And this we knock down the HMGB1 in the X minus system. It doesn't affect the replication that much. We make a similar amount of cccDNA, but the cccDNA transcription is significantly upregulated after knocking down HMG1. And you can also see the epigenetic markers associated with that also changed to the positive markers, but the regressive marker is reduced. And this also shows that in the infection model, knock down HMG1 also can epigenetically upregulate cccDNA transcription, either the wild type or the X minus virus. And what's the mechanism? We have some preliminary data show that the HBX and HM can interact with each other by the co-epi study and the polarization study. And also the binding of the HMGB1 is X also changes the HMGB1 cellular localization. So in originally HMGB1 is predominantly in the nucleus, but after HBX binding, some of the HMGB1 actually move into the cytoplasm. So that could be two possibilities, that can for HBX to antagonize HMG1 by directly binding or changes subcellular localization. So you lose the chance to bind to the CCC. So this is the model for the current. This one we just published just online this week. This paper to just show that HBX is a viral factor that antagonizes HMG1 to epigenetically upregulate CCC transcription. So to summarize what we have done recently on the cccDNA biology, so the HBV rcDNA is the damaged DNA. It processes damage-like signals, and the DNA repair machinery is required for cccDNA formation. And the viral polymerase and the RNA primer are removed from the cytoplasm of the rcDNA, which generates this DPrcDNA, and so far, but we still don't know the enzyme remove the protein from it. And the nuclear rcDNA can be recognized by DNA damage sensors, like the UVDDD and also HER that's identified by Shenk host site. So those are the early factors involved in cccDNA formation to initiate the DNA repair. And the cccDNA chromosome undergoes epigenetic regulation, and the HBX protein maintain the transcription activity of cccDNA by removing the host epigenetic silencers like HMG1 or the SNAP6 information. And so we think HBX and cccDNA epigenetics can be an antiviral target for treatment of chronic hepatitis B. So the question is that if we want to eliminate cccDNA, that's maybe really difficult because it is as just a DNA in the host cell. But epigenetic silence cccDNA transcription activity, which is the only activity of cccDNA, that could be managed, and lead to a functional cure of HBV. So that's my plan B. Instead of eliminating cccDNA, maybe difficult, this one could be a feasible approach. But sorry, team, we still don't have a way to cure hepatitis B or eliminate cccDNA. So please stay. Okay. So I just want to thank the people in my lab and my collaborators, including people in this room, of course, who I collaborated with. And I mentioned that I have moved to Drexel, Doylestown, to Indiana, and to Pittsburgh. I'm still working on hepatitis B. I'm committed to hepatitis B, and hope everybody can work together to find a cure for hepatitis. And I want to share some moments with the team. I don't have many pictures with the team, unfortunately, but these are some I can find. So this one is in the 2010 HB meeting in Taipei. So we just, hungry people on the HB meeting, walk on the street, and we randomly find a restaurant, and the food tended to be pretty good. Look at deep space. It seems like you didn't like the food that much. Right, and this is Richard. That's my first, like a visiting scientist or a PhD student from China. So he joined here and worked with me and Endowed. We published many papers. Tim left him. Very much. That's the reason when he left the foundation. And this is 2011. That's not me. That's you. And this picture, you have seen that many times in the workshop. I always use this one to show, my presence in the Hepatitis B Foundation, and along with many prominent scientists in the HB field. And this is a dinner with a bunch of Asian scientists, basically Chinese scientists. So I think I really appreciate that because we always promote international collaboration and international scientists to come here. And he had a program called Scientists Without a Border. You can see there are a lot of Asian face in this building. So that's a big achievement and really help the field to produce young scientists and a new generation for that research. We appreciate it. Thank you so much. So that's the end of my talk.
Do we have questions for Haitan?
Wonderful talk. It's nucleotide system repair is the only mechanism that is required, or do the other mechanisms of DNA repair collaborate? And might the combination of the DNA repair inhibitors help eliminate?
Yeah, that's a good question. I mentioned that there's overlapping functions of the DNA repair machine, even for hosts, not only for hepatitis, for the host. Right? So the virus actually is smart, not as smart, but smart. To utilize this kind of overlapping function for its own use. Talk about using so far, all the DNA repair factors involved in CCD formation is not blocked. No one can completely block CCD formation. That's a clear sign for redundant activity in the repair.
For knocking out each of the factors.
For one factor. Yeah. For the whole pathway, we cannot do that so far because these may also cause cell death. That also leads to the question: we want to target CCC in formation as a therapeutic. So that could have some toxicity issue. So that's basically, I think it's more like a basic research. But if you use that for short time period treatment with other drugs, that may be helpful.
And I know it's for the work that we've done in Biomarkers, that it is small. That makes it so it's more susceptible to other that would kill the whole cell.
That could be a selective elimination.
It doesn't propagate on its own, right? So what propagates CCC?
CCD doesn't replicate.
Right.
You only convert it from the RCD.
So you could selectively, perhaps selectively kill those cells.
Yeah, that's a good part. Yeah, Evan.
Actually, also do something on the action to be like that's fine to post from.
Right. But we're still working on that. We're doing the PhD. And the data is still cooking. Yeah.
That's some more general question. So you can under that, Joe and other labs have established the basis for the CCD also this and those are for epigenetic regulation. So over the years, have you got any clues? Is there a specific way to specifically CCD not targeting the host? Do we have any clue so far?
Right. But so far, all the epigenetic information we can use from the host study part, right? Basically, just borrow those into the CCC study. So far, you cannot just completely distinguish. It seems like you target the CCC, and probably also target the host part. But as I mentioned earlier, so this could be a short time treatment. You can quickly, rapidly silence CCC and still the host universality is permanent. That's good. But we have other proof.
The jacket that team wear in 2011 is the
This tie is already 10-year-old. That's your question?
Thank you, Mike. Thank you to all of the speakers. This has been a terrific symposium. Just wonderful to hear how all of you have worked together with Tim and created just a marvelous environment. It's been so incredibly productive. So thank you. This has been a lot of fun. So now I'd like to invite Shari Cohen, the president of the Hepatitis B Foundation, and Lou Casa, the president of the PA Biotech Center, to come and take over the program.
So much. Is that right? Thank you very much. I do have something to read from a guest who couldn't be here, but before we move forward, Tim, there's a word in Hebrew for people who dedicate their lives to the service of mankind, for people who are kind and generous and love others and constantly give. And it's called a mensch. And from what I know of you, working with you and living beside you and raising our kids together for the past 25 years, and everything we hear today, you are a mensch in the true sense of the word. So I have the honor of reading the Timposium, which was written by Dr. Harvey Alter, whom you all know and love. He was a member of the Hepatitis B Foundation and also won the Nobel Prize. The Hepatitis B Foundation is way more important, of course, than winning the Nobel Prize. He's very sorry he couldn't be here today, but he is out of town. He's out of the country, actually. But here's the Timposium. When I was a child, I played with blocks. When I was a teen, I played on the block. As an adult, I traveled with the blocks. I am proud to be a chip off the Tim block. His pace is tiring. His thoughts can be mystifying, but his visions are aspiring, and he is always inspiring. I could not be more admiring. He is my mentor, my HBV center, a biotech renter, a techno inventor, and amidst this all, he couldn't be gentler. For his praises, there is no dissenter. Who knows what life will bring around the next bend, but I am privileged to know that Tim will always be my close friend, Harvey Alter. So we have a couple of presentations to make for you. The first is something that we have all put together. So for those of you who know, Tim pushes swag on everyone. You got the ties, you got the livers, you got the mugs, you got the hats. I don't know that you have too many, and so we put together a basket of swag for you that I know Joan's not even gonna let you take home, but it's a bag of Hep B Foundation and Blumberg Institute and Biotech Center swag. And then this is a little bit more serious. We wanted to get you something that you could have on you and keep and remember us. And so there's a watch for you. And we had it engraved with your initials and then March 27th, 1991, which is the date of the incorporation of the Hepatitis B Foundation. So hopefully we're in good health. And thank you.
Thank you.
Two more things that we're not talking about. The price is right there for a second. There were two more honors, tributes to Tim. First of all, I want to mention everyone, a few years ago, we had a sign outside the Biotech Center. We walk up and it would say, "Welcome to the Pennsylvania Biotech Center," co-founded by the Hepatitis B Foundation, and partner I will not name due to legal obligations at this time, and probably some confidentiality. And one of our attorneys, Lauren, the chair of the ABC, is in our audience. That bothered me, and somehow, a few years ago, it mysteriously disappeared. We are a 150,000 square foot campus, or 14 acres. You lose things every once in a while. But today, on this day Timposium, that will live in history, infamy, we have a way to make things right. Dr. Block came up with the idea of the Biotech Center, created the Biotech Center. He fostered this environment for research and culture and synergy. And I think today it will be with great honor. You want to come over here? We are going to name our campus the Timothy M. Block Research Campus. So everyone, if you have your packet, get ready for a second here. Tim, you impacted so many people's lives with your leadership, your vision, your intellect. You can tell by the witnesses of testimony today, you've really changed lives. And as a symbolic tribute, I'd like everybody to take out their tin case. Tim, I want you to come up here. Make sure you have your face. So this, I know you can't hear the microphone, but this is our way of showing how you impacted us. And we want to take this and unleash more Tim Blocks on the world. So thank you so much. And I guess with that, we see this every day.
I don't know that is what we really want.
So with that, the man of the hour, Dr. Timothy Block.
Thank you. Thank you. I've heard this committee prepare anything, so I would like to just say, because I have to be careful, I don't want to say anything wrong or illegal. So I'm going to, so I'll be careful. But a couple things I do want to say. One, first of all, I appreciate everyone coming. Some of you from long distances, and of course the great science that I've heard from the people that I've worked with, that their generosity in crediting me or attributing so much to me, it's really over the top. And I very much appreciate it. I can't say enough. I'll say a couple things for some of the people who didn't say, talk a little bit about what they did. Ellen Feynman, I'm sorry, I don't remember his last name. Ellen got married since it has almost 35 years. But Ellen, by the way, her work, not with me, but with Harry Robinson, the first DNA vaccine work to be reported, which is a big thing. Ellen went on to be chair of the department at the University of Massachusetts. So that, and Satish Chandran's here too, I saw him come in. And Satish, if you still, yeah, Satish, again, nothing to do with me, but while at the center with Kathy Kinchak, developed the first siRNA construct they call post-transcriptional silencing or something, I can't remember, but it wasn't called siRNA at the time, but they actually did the first work. And so, and they didn't talk, but I also want to mention, of course, I'm overwhelmed with renaming the campus. That's an enormous honor. And, you know, my own organization doesn't have Joan up there because, of course, Joan, you know, everything I've done has been with Joan. All this makes me not want to leave. Now, don't get scared. I'm sorry, I am, but it really is such a thrill to hear all this, to see all this. And you know, I come home sometimes and play the government. Oh, I'll never do that. I can't say that anymore. And having the meeting kicked off by Raymond Dweck and Tom Shenk, two big giants in the field, of course, is very humbling. And Tom, you did a great job. I went to a couple of weeks ago. Joan and I were had really the honor to be at Tom Shenk's retirement party, going way away, retirement party, and I learned a lot about that. And one of his former grad students or postdocs got up and said when we got the notice, they thought that it was a memorial service. I'm saying it's kind of the same thing, that you get to hear all the great things that he said at a memorial service. But that guy said, he's not a kid anymore, you know, he said, and so he thought that he was so relieved to hear that Tom wasn't dead that he got there. But then, maybe you didn't know what he said. He said when he got there, he said, Tom almost looks lifelike. He didn't know whether. It is, I kind of feel the same way. I'm getting the real thrill. I hope you all get the chance to have this done for you. There are all these nice things said about you where you're still alive, because when you reach my age, you go to an increasing number of memorial services, and you're hearing all this, yeah, that would be nice to actually stay to a person if they were alive. Well, I definitely want to also recognize Megan Pierce. I don't know if she's Megan. And Gene Holmes, who worked on this, and everyone has come together, not just for this event, but to make the Hepatitis B Foundation and the Blumberg Institute and Biotech Center the successes they are. Obviously, these places are enormous successes, and I appreciate the recognition you've given me, but really, if there ever was, and we're still, as I said at our faculty meeting recently, we're very early in the story of these organizations. So everybody in the room here has contributed as part of the beginning and part of the success. I don't want to give up the mic. I'm looking around. I'm just looking around at the decades of my colleagues, trainees, and people who come in, and I'm just so proud of everything they've been able to do, and really humbled by their willingness to recognize my role. And it's been an enormous pleasure, a lot of fun. And I am retiring, I'm leaving. The leadership of the organization is put in tremendous hands. And Lou and Sherry are not presidents yet, but another week. but my goodness, the place is in really, really great hands. And I appreciate all you said and done and have continued to do to make this place what it's become. We've seen the growth of these organizations with their help and their effort, and we're going to be in great shape. So I know the party goes on. It's a little like you can see I don't want to go away, but I have to. And I should also say I appreciate it all from heart.